In this Facebook post, the title is Researchers Find Cancer Undo Button, Turning Tumor Cells Back into Normal Cells. One of the exciting things about a lot of the new research that's coming to fruition is with all this genomics and transcriptomics type of capabilities, we're starting to find sort of these checkpoints or different convergence points where essentially there are critical genes that if we could somehow influence, it may totally disrupt this whole carcinogenesis or oncogenesis type of pathways. And so one of the things that kind of caught my attention was this post is that basically a scientist in Korea identified some genes that potentially, if they could knock out or block, so basically they take the gene out of commission, that the cancer cells reverted back to normal cells. And it's pretty fascinating research. The website, or I should say the paper, if you this is part of a research group called KAIST, KAIST, but it basically says KAIST develops foundational technology to revert cancer cells to normal cells. You got to dig around a little bit, and then you could find the actual paper that the data was published in. It's a journal called Advanced Science, and the title of the paper is Control of Cellular Differentiation Trajectories for Cancer Reversion. And so basically, you read through this, and there's a lot of technical details. And so what I thought is, okay, what if I just look at like what's some of the limitations? Because it sounds very, very promising. What's one of the problems with this, like right now? So if you have cancer, you're gonna have time to wait for this to come to fruition. And some of the things that came up when I put this into an AI model is that the scalability. So the particular Boolean network modeling approach these guys use may face scalability issues. The other issue is that the inferred gene regulatory networks might be incomplete. And the third one is that there are time point assumptions. So the methods used, they assumed irregular time points and inferring the regulation logics. And this may not actually reflect the continuous nature of biological processes. So basically, what these guys did in the study may not reflect what all cancer cells are doing. And so, as exciting as it is to think, hey, we could take a cancer cell and revert it back to a normal cell, there's quite a bit more research that still needs to be done before this can be ready to, start using and rolling out for everyone that has cancer. So I hope this was informative and you got a chance to get a different perspective on, some of this exciting research.